Hey everyone, my name is Giff Ransom. I'm a 3D graphics programmer by trade. I work in virtual reality, and I am fascinated by molecular biology. So, in sort of the programming Twitter world, there was an article going around right around Christmas, basically walking through the source code of the new Pfizer vaccine for COVID-19. And it went around, like programmers like me saw it, read the article, and thought it was about the coolest thing they had ever read. It was written by a guy named Bert Hubert. He's a programmer like myself and has way more biological knowledge than I do. And I was just fascinated to read it. And it's so cool that I really think the general public ought to be able to understand it too. And there's a little bit of specialized knowledge there. And so I thought I'd just take it on myself to try to walk through his article and explain it to people who don't have a lot of knowledge of what all this stuff is. And I'll try to cover it as simply as I can to give you a sense of what's going on, because I think the more people know what this thing is actually doing, I think just the more excited they're going to be about the times that we're living in. There's been a lot of negativity, a lot of just people feeling like, oh, this is a bad time to be alive. And there's a lot of amazing stuff happening right now, a lot of ways in which it's a really great time to be alive. So I want to share a little bit of optimism and excitement and wonder with y'all. So he starts the article talking about what does it mean to look at the source code of a vaccine. I mean, a vaccine is a gel that's put in a syringe and injected in your arm. How do you look at the source code for that? And the answer is that vaccines in the past have had a lot of different little maybe parts of a virus or a dead virus or a really wounded virus to put into your body so that your immune system can learn how to fight it without it actually being dangerous to you. But what these new kind of vaccines, these mRNA vaccines are, is instead of actually putting any virus into you, it sends a little blueprint called mRNA into your body to be sent to your cells' factories, which are then going to produce this little part of the virus for your body to practice against. And so that's different. So what it is actually giving your body is source code, is information. And that's different than giving it, physical things. So this is a really new technique, a new process. And because it's giving information, we can actually look at it. And he points out here, this is the first 500 characters of it. There's about 4,200 characters total on this thing. It fits on 2 pages if you print it out. And this article is actually looking at what does this information contain. All right, so here's where he starts giving the background that you might need, just a little bit of the biology you might need to understand what's going on here. Now, here's where I'm going to stop and try to explain, maybe in a little more depth and a little more basically, just what modern biology is all about. What actually we understand a cell to be that may be different than what you remember in your high school biology textbook. Certainly what I remember. So, buckle your seatbelts because this is going to blow your mind. If it doesn't, you're not paying attention. Okay, so on the internet now, there are some fantastic videos and computer-generated things that show us a glimpse of what's going on inside the cell. And the cell, so far from how we may have been brought up to think about it, this little bag of goo with a few identifiable parts or something, it's not that at all. It's more on the scale of complexity of, say, a modern city and all of the infrastructure that it takes to run a modern city. So this fly-through that we're looking at is basically going into the superstructure of the cell, getting in and looking at all the little parts on the border. There's power generation that has to happen. There's a lot of transfer of parts in and out. There's transferring of information from the control center to the little factories to produce machine parts and put those machine parts together into working machines. And it's really amazing. It's a vast, almost universe inside that's too small for us to even see. So this guy here, this is a protein transport system. It's got this little walker thing that walks along these roads, basically, and at the center there is the nucleus. So let's look into a little bit more about like what is happening here. So the heart of all of this is what we call a protein. And a protein is just a little machine part. There are, just any number of different types of them, and they're just little 3D pieces that do stuff and that fit together to make little machines. Here's an example of one of those machines. This is called ATP synthase, and it's a little power turbine. And it's composed of several protein parts put together. And you got part of it that's twisting, that's powered by some electrical charges. And this other part up top, these three proteins that do this mechanical clamping. And what they're actually doing is they're putting two little molecules together to make a little battery molecule. And your body uses those all the time, these little one-shot batteries. So you see these different proteins put together for this power turbine, and its job is to power your cell. Here you see a huge number of these little turbines churning out the batteries at breakneck speed. There's countless numbers of them in every one of your cells powering it. Here's another machine. It uses multiple proteins, and its job is to copy your DNA. So you have the DNA strand running through there, and it gets split. The double helix gets split into two halves, and they each go into a little subunit where the other half is completed. So you have two strands. And there's something interesting going on here with this machine, because I guess the part that actually completes the double helix only works one way. So it actually has to double back and basically copy it in reverse. But again, here you see multiple proteins come together to one assembly. So a protein is a machine part. But where do these proteins come from? Let's look at that. So to make a protein, we have to start by copying the section of the DNA that tells us the schematic for building it. And so that's what's happening here. The DNA is being kept very safe in the nucleus of the cell. It's wound up tightly and protected. So what we have to do is make a blueprint, make a copy. And that's what this molecular machine is doing. It's opening the double helix so that it can make a copy, base by base, and then that copy, that blueprint, which we call RNA, is going to go outside of the nucleus of the cell into one of the protein factories and show it how to build it. Okay, our blueprint has arrived at the protein factory, called a ribosome, and we have to read it bit by bit to see how to generate the protein. A protein is composed of these Lego beads that we call amino acids that link up into a chain and then fold up into a 3D shape. And because there's 20 different types of amino acids and only 4 different bases, we have to read them in groups of 3, because one base can only generate information 1 of 4, 2 you can get 1 of 16, so if you get 3 you can get 20 and more. So it goes in groups of 3 over and over and over and links it up into the chain of these Lego beads, and they fold together. Let's look at that in a little more detail. So here in this more detailed animation, we can see as these little Lego beads come out, they don't stay just a long chain. They actually fold up and snap at very specific parts. So depending on which of these 20 different bead types you have, it might have a connector to the left or a half turn or a connector on both sides or whatever. And they snap together at specific points. And what that ends up meaning is that depending on the sequence, it'll define that 2D sequence will define a specific 3D shape. And so then you have at the very end of this a protein of a very specific 3D shape with little charges or whatever at different points, and that'll serve its function in a molecular machine. So we've gone from information in the DNA to a completed protein. Okay, so why do we care so much about proteins? Well, it turns out it's not just our body that's made of proteins, so are viruses. And the coronavirus has this really distinctive spike protein that everybody's seen. And what the spike protein ends up doing is serving as a key to plug into our cell's portals, open it up, let the virus in, and the virus has a bunch of machinery that it plugs into our cell's factories and basically turns our cell into a virus factory, which is really nasty. But that spike protein is a weakness because it has a very distinctive shape, and we've actually been able to figure out exactly the sequence to make one of these spike proteins. And so the idea is that if we can get our body to make a bunch of free-floating spike proteins, our immune system can build a mold to it, it can figure out how to attack it, and then we'll be able to attack the coronavirus directly. We'll have immunity. So that's the game plan. So here we have on this document that I'm showing you, the entire mRNA sequence for the Pfizer vaccine. Like I said, it takes about 2 pages. And this is the sequence that Bert Hubert's article goes through bit by bit and explains what each section is doing. It's all kind of open source for all of us to look at it. So let's take a look. So we start out with a table of contents, basically, to say what all the different parts of this schematic, this blueprint, what they mean. So we start off with a 2-character cap, and then there's the header here. And then this middle area is the actual code for the protein itself with a little tag in front. And then there's a footer, and then there's a tail. And he walks through each section and explains what each of them do. So let's start with the little cap at the very beginning. There's these 2 letters, GA, these 2 nucleotides. And he says they're slightly chemically different than the rest. It's just kind of an extra signal. But the point is that if it doesn't start with a GA, the ribosomes, the factories, are not going to touch it, because this is a little signal at the very beginning that says this is coming from the nucleus of the cell. This is something you need to use to make a protein. Now, actually, the mRNA in the vaccine didn't come from the nucleus of your cell. It came from the vaccine, but we don't want to tell the factories that. We want them to make the spike protein. So that's how we start. So we got past the cap, and the next section here is the header. And here's where we run into something really weird. So the different base types in RNA are G, A, C, and U. But he points out, so why are they using this Greek letter psi, this like U with a line through it? And the reason they're using that is that the actual chemical composition for the base is slightly different than a normal U that they're using for this. And they discovered this through some research like 3 years ago, pretty recent. And they found that if they alter the chemical composition slightly, that all the cell's machinery for copying RNA and for decoding it to make proteins and everything, that all works fine. But the part of our immune system that's really, really trying to kill any RNA that looks slightly suspicious for good reason, to fight viruses and whatnot, that misses, somehow it misses it when you use that cyan. So the goal is to get the RNA into your body, into your cells, so it actually does make the spikes. So we're actually at this stage of this process actually trying to bypass our immune system. So it's kind of a cool trick, and amazing that we discovered it. Now, the one thing that you can ask is, well, couldn't that be dangerous using that? You know, if the immune system isn't picking up on it, couldn't this be used for nefarious purposes? And it might, but the interesting thing about it is that if you were to ever make a copy of an RNA sequence that had all these little psi bases, these modified U's, it would copy it using your body's normal U's, in which case that copy would be immediately attacked if it ran into our immune system trying to fight it. So the idea is that it's kind of a one-use thing. Once it deteriorates, it's gone. So it's kind of a neat trick to get these things into our cells, bypass the initial defenses, so that we can start using the spike protein. Now, what does this header actually do? The big information we know that's encoded in the header is when do you need to make this protein and how much of it do you want to make? And what we've done is looked at other blueprints that our cell uses for different proteins and whatnot that it normally does it. And we've picked the one that says, make a lot of it and make it right now. So that's basically what it says. Okay, we've gotten through the header. Now we're going to get to the tag. So this is a part of the actual protein. We're getting to the part where it's actually giving code for the amino acid sequence, the little Lego beads that get folded in the protein. And at the very beginning, we have a tag, and it's not really going to be part of the final protein. It's more a list of instructions for when you finish this protein, what do we do with it? Where am I going to take it? Where am I going to ship it to? And the tag, the address label says, send it right out of the cell. Okay, so then Bert goes on to compare the actual sequence for making the spike protein that the vaccine has with the sequence that the virus uses to make that same spike protein. And he notices that there's big differences right off the bat. As you can compare the virus with the vaccine, the second base has a difference in the letter, and so does the third, so does the fourth. And he's wondering what's going on here. So to understand this, we got to go back to what we saw earlier, that if you're going to encode for something that can be 20 different things, as there are 20 different types of amino acids, you can't do it with one base because there are only 4 different base types. So that could only give you information about 1 of 4 of something. If you have 2 of them, you get 4 times 4. With 2 bases, you could have 1 of 16 of something. If you have 3, you could have 1 of 64. Well, we don't need 64, we just need 20. So how does that work in practice? As it turns out, what that means is there's redundancy. There's like 3 or 4 different codes that match to the same amino acid. And so what they've done is they've said, if we want to kind of even out the letters, or in some cases they research that it actually ends up being better to have more C's and G's than the others. Since it's going to make the same protein in the end, let's just use the redundant sequence that has more C's and G's. And he actually found only one there that didn't result in an additional C and G, and he asked the crowd, what's going on here? And they said, well, in that case, when you have a couple of these bases together, if you had it in that sequence, it might make a little bit of a kink, like you get in a hose sometime. And if you swap this out with another one that will do the same thing, it sort of breaks the kink. So they're looking at all these things as they're creating code for this blueprint. So then Bert looks at the whole sequence for actually building the spike. And he sees a lot of these discrepancies that we talked about before to make it a little more efficient. But then there are some actual changes in the structure of the spike. And he says, why is that? Well, as it turns out, when you have these spikes, normally they're attached to the skin of the virus. So you can see all the little spikes around it. But the spike we're making is going to be free floating. Well, why does that matter? Well, it turns out that if you have a spike that's just free floating, it kind of collapses. And that's not going to look the same as one of the spikes that are actually attached to the virus. And that's a problem because we want our body to practice against something that's as much like the virus spike as we possibly can. So what they had to do is they had to engineer, okay, at the part where it attaches to the base, how do we get this thing to stay rigid even when it's free floating? And they figured it out. They figured out how to actually create a rigid free floating spike. So they made that change. Now we get to the end of the protein coding sequence. And there's a little codon that its job is just says, stop coding the protein, we're done. And it's kind of funny. It looks like we put 2 of them in there, and just maybe just to be really sure. He says we're not really sure why they needed 2, but they used 2. And then after that comes the footer, the 3' untranslated region. And here Bert says that actually we kind of don't know what this part does. And so we're just using the ones that already exist, and so that it can be like the other ones. And we found that there are particular ones that seem to, again, get your body to make more of these things. So we're using those. But this is still a bit of a mystery to us. And then finally, we get to the very end. And it's just, I kind of made a joke. It's kind of how we all feel about this year. Just nothing but A's all at the end. And what are those for? Well, we found out at the very tail of the RNA sequence of the blueprint, eventually a bunch of wear and tear happens and it starts losing bases at the end. And they found that a lot of A's, about 120 of them, is kind of the optimum amount to keep this blueprint around as long as it can. And then eventually it'll deteriorate, and that'll be that. Your body will quit making these spikes. So then Bert summarizes it all. What do we have here on these 2 pages of information? So as we saw, we've got the cap to make it look like all the other mRNA that comes from the nucleus. We've got a header that's known and been successful and optimized for generating a lot of that protein. We've got the little tag that's going to be on the protein to tell it where to go when it's done. Then we have the actual code for the spike protein, which we've altered to make it rigid, even when it's free floating. And then we've got a footer, which we're, again, not really sure what it does, but we're using the other footers from other mRNA sequences that our body uses that tend to work pretty well. And then finally, there's the tail that tries to let it stick around as long as we can. And that's it. That's what is being injected inside you. It's this schematic, this blueprint that goes straight to your factories and starts making the spikes. So my takeaway from all of this is to have a little bit of gratitude to set against the suffering and the frustration and even the anger with all that we've been through in the last year with the virus and the measures that were taking place and the societal conflicts around them. To be able to look inside our own bodies and the universe that's unfolding within them, and to actually start to be able to help on this scale with a little bit of real precision is amazing. And I think even amidst all the suffering, we can look at that and be grateful to God to be alive at this time. And so I want to share a little bit of my own wonder and awe at all of this with y'all, and I hope it's uplifting.